Hello friends, welcome to Dr. Sai Physiology Academy, DOPA for short. This is the place where we make the learning of physiology easy, exciting, and effective. Thank you for joining me. Alright, so today we are going to be doing the part 2 of the introduction to renal physiology with special reference to body fluids. Always remember that learning how the body functions can teach you about vital health lessons, life lessons, and even management lessons. Okay, so let's just do a brief recap. We talked about how the body is like a manufacturing plant, and in the process of working, there are a lot of waste products, and the kidney is the chief excretory organ of the body, and it uses a lot of water to excrete those waste products. So that's why we are starting with water, the body water, understanding how it is distributed. So we said that the total body water, total TBW of the body, the average person, is about 42 liters. This 42 liters is in a 70 kg man. That's just like the standard physiological weight of an adult man, okay? 70 kg. So in a 70 kg man, 60% of this, of 70 kg, is 42 kg. And we said 42 kg is equal to 42 liters, all right? So the total body water is further divided into two major compartments: the intracellular fluid compartment and the extracellular fluid compartment, outside the cell, inside the cell. And out of this 42 liters, this one takes 28 liters. The ECF takes half of it, 14 liters. Okay, so the extracellular fluid compartment is now further divided into two. This one is called interstitial fluid compartment. That's the one that surrounds the cells. And this one is the plasma, the one inside the blood vessels, also known as intravascular. And we said that this one, out of 14 liters, takes about 11 liters. This one takes about just 3 liters. So 1, 2, 3, 4, 5. We've mentioned 5 different body fluid compartments with 5 figures, 5 volumes. How were these volumes measured? What was the method they used? Because intracellular fluid compartment has to do with the volume of fluid in all of the trillions of cells in the body. So learning these methods can teach us a lot of things. So, and that special method is called the Indicator-Dilution Technique or method. All right. So it means that there is an indicator. You also call it a marker. So that marker is introduced into the body. It's injected into the body and is allowed some time to mix with the body fluids. And then you now collect and withdraw a sample and measure it. But for us to understand it better, let me illustrate it like this. Let's assume you have a glass of water. You fill it to 1/4 level, okay? Then you take a tablespoon of milk, put it inside, stir it, mix it very well, keep it, and take another glass of water, fill it to the brim. Take the same 1 tablespoon of milk, put it inside, mix it. You taste one, taste the other one. Do they taste exactly the same thing? The answer is no. One tastes, it's more concentrated, it's thicker. The other one is light, it's more dilute. So the taste is not the same. Your tongue is able to detect that there is a difference. So the same thing happens. So I've mentioned three things. I mentioned amount, I mentioned volume of water, okay? Volume. And I also mentioned concentration. Concentration, okay? So what is the relationship between all of this? You also know that when you increase that tablespoon of milk to 3, the concentration generally increases. So the relationship between amount and concentration is direct. You increase this one increases. Okay? So, but volume, the relationship between volume and concentration is opposite. When you increase the volume of water, the concentration reduces. So volume is inversely related to concentration. Okay, they are opposite. You can also put concentration here. They are the same thing. So now, let's now make a new equation. So concentration is equal to amount divided by volume. Inversely proportional to this, directly proportional to this. Okay, so if we cross multiply, because we're interested in volume, volume will give us amount over concentration. Okay, so this is the formula we're going to be using to know the volumes when we want to measure them. All right, so let's go back to the indicator and the markers we're talking about. So now you introduce a marker. Let's say, for example, you want to know the volume of this compartment. It means that that marker has to remain in that compartment. It doesn't have to move or shift to any other compartment, or else you get wrong results. So one of the properties of the substance we are going to be introducing is that it should remain in the compartment that you are interested in. Okay? It should remain in that compartment. Another property of a very good marker or indicator is that it should not be toxic. That is very basic. It should be non-toxic. Then another one is that it should be evenly distributed. It means that there should not be some places that are more dilute than the other. Because when you are collecting the sample to measure, if you collect from a place that is more dilute, you get a wrong result. Or you collect from a place that is more concentrated. So it has to be evenly distributed. We have even distribution. Okay? So another property that this marker, another criteria it has to fulfill, is that it should not be easily excreted. So when you inject that substance into the body, maybe 5 minutes later, more than half of it has been excreted, you will get wrong result. So it should not be easily excreted. Then another major, very, very important property and criteria it needs to fulfill is that it should not be metabolized. It simply means it should not be transformed into something else. Maybe some enzymes in the body will act on it and change it into another substance. You will miss what you're trying to get. So it should not be metabolized by the body. Okay? There's some other little properties, but these are the major ones. Okay? So that is what that marker or indicator, it has to fulfill. So, and you know that these different compartments, that you see mentioned in the part 1, that there is a membrane that separates them. Okay? The intracellular compartment is separated from extracellular by the cell membrane. Then the interstitial fluid compartment is separated from the intravascular compartment through the capillary membrane. We'll talk more about that. So the next thing we're going to be looking at is the substances that are used to measure each and every one of these compartments, because it's not the same. They are in different compartments. Each of these compartments, you need a unique substance or marker to measure them. But the thing you ask yourself, can you gain access to each and every one of these compartments? Gain access to this, because you say it should remain in that compartment. That means you need to gain access specially to only that compartment you want to measure, and it should not move to another compartment. The answer is that no, we cannot gain access to these compartments individually. The only compartment that we can gain access to is the intravascular, the plasma. The plasma is located in the vessel. So how do you now do the rest? It means that there is a relationship and a connection between all of these compartments. Let's just visit it a little so that you understand how it works. For example, this is the heart. Okay? Then from the heart, you have arteries. So let's use a red biro to indicate arteries. Okay? The heart gives out arteries, big arteries, and as it goes down, becomes smaller and smaller and smaller till it gets to the capillaries. Okay? So the capillaries are now involved in exchange within the cells and tissues. So here you have different cells making a tissue. So in between these cells, like you know, is the interstitial fluid, also known as tissue fluid. Okay? So the capillary is located in between the cells also, just like the interstitial fluid. It's located in between. All right? So the next thing you now have is that after being in this tissue, the capillaries will now rejoin to form venules and then veins, which will now go back to the heart. So if you look at this diagram, the diagram is not so perfect, but it will help you understand something. This system is a closed system, very closed. Okay, from the heart is connected to the arteries, to all the tissues. This is just a representation of different tissues and organs in the body. Okay, so anything that enters here will go to all the body. Anything that enters the veins will go to every part of the body. They are all connected. Okay, so now within this place, you find that you have the interstitial fluid. The interstitial fluid gains access into the capillaries, which now enters the general circulation. Then the interstitial fluid is in direct contact with each cell. So there's exchange of material between cell and interstitial fluid. Then from interstitial fluid to the capillaries, which is now in a vessel. Okay? So that is the connection. Inside the cell, ICF inside the cell has direct connection with the extracellular fluid. The membrane separating them is the cell membrane. Then the interstitial fluid has direct connection with the intravascular fluid or plasma, and the membrane separating them is the capillary membrane at this level. So it enters into so that is the connection. So it means that if you introduce any substance into the vein, it can get to all the compartments. And we use the vein because the vein is easily accessible. There are some people, when you look at their hands, you see a lot of veins showing. So the veins are very easy to access. So that is the only place we can gain access. All right? So when we gain access to this one, we gain access to all the rest All right, so let's go into each and every. We'll start with the total body water. Let's clean this. So you ask yourself, how do you measure the total body water? The total body water means every compartment, okay? And the substance that can be distributed to every compartment, it can penetrate everywhere, is water itself, okay? So but that water is a special water we're going to be using. It's called heavy water. So for the TBW, total body water, what we use is heavy water. It's also called deuterium. Deuterium. Okay? It's written as this. Alright? So that is heavy water. It's different from normal water. The difference is that the hydrogen here has one neutron and one proton. The normal hydrogen has just one proton. So that's why it's heavier. So it's an isotope of normal hydrogen. So it's a special. Another thing you can use is called tritiated water, also known as tritium. That one is written like this. So this one is known as radioactive water. Okay, they are different from the normal water. That's why that's why it's called a marker. So it can be easily identified and separated from the normal, but it has similar properties. So it can still penetrate. So to measure the total body water, we use these two substances mainly, radioactive water or heavy water. So we introduce a known amount into the veins and we give it some time so that it will mix, it will go everywhere to mix evenly. And after some time, we'll withdraw a certain amount and measure the concentration. Okay, so we now do this. Volume equals amount over concentration. So the amount we introduce is known. Then when we withdraw, we measure the concentration. So this is known, this is known. So using this together, we'll now get the volume. So that's how 42 liters was gotten. Okay, so that is total body water. Now, the next one we'll look at is number 1. Number 2, extracellular fluid compartment. Okay. Now, ICF, we are not going into that because it's difficult. Remember that we said that if you want to measure, it must remain in that compartment. So, we cannot introduce through the plasma, and this is the only place you can introduce a substance. So that means it's impossible. Because if you introduce it here, it has already entered another compartment. It will have to go through like this and enter interstitial, enter here. So it's impossible. So we are measuring this. Okay? Because this one comprises of this and this. So this one goes to the ECF. But it must remain in the ECF. It cannot cross. So we are going to take a substance that is incapable of crossing the cell membrane to gain access into the intracellular fluid. Okay, that substance is radioactive sodium. Radioactive so that it can differentiate it from the normal sodium in your body. So we use radioactive sodium. Okay, so generally sodium is restricted to the extracellular fluid compartment, though little sodium can enter into the intracellular, but it's negligible. So that's why we use that. So when we introduce a known amount of this radioactive sodium, it will distribute only in the ECF. Very little enters the ICF. Okay? So, and then we take the sample back and measure the concentration. Then we get the volume. Alright? So that is this. So, we've gotten this. We've gotten this. But we cannot get this directly. So what do you think is the logical thing to do? Since this is a sum of this plus this, it means that to get this is this minus this. Okay? So ICF equal to total body water minus ECF. Okay? So that's how you get the ICF indirectly. Since you can get this, you can get this. So you just do the subtraction and you get this. All right? So the next one I'm going to be looking at is the plasma. Why? We cannot also get the interstitial fluid compartment. Because if you go here, you have to pass through the plasma to gain access to this one. So it has already spoiled that law that it has to remain only in this compartment. Okay? But if it remains only in this compartment, you can't get it back. To get it back, it must pass into this plasma. This is the only place you can gain access. So that's why this one and this one is impossible to measure them. So let's get this. So to get this, we need a substance that can only remain in the blood vessel. It cannot cross the capillary membrane. And what is that substance? It's called albumin. So the plasma, you use albumin that is labeled with a radioactive substance, radioactive iodine usually. So we use albumin that is labeled with iodine. Okay? Radioactive. So the radioactivity is to differentiate it from the normal, because your body has albumin normally. Okay? So it's to differentiate it from the normal albumin in the body. So albumin cannot cross the capillary membrane into the interstitial fluid. So it remains only within the plasma, the intravascular space. Alright? So when you do that, it's distributed all over. Then you now collect it and measure the concentration. Then you get the volume. So the same thing we did here, we also do it here. Since we know the ECF now, and we know the plasma, and ECF is the sum of this and this. So what do you think the ICF will be? The ISF will now be ECF minus plasma. All right? So we've gotten the 5. So 3 can be gotten directly. 2 are gotten indirectly. So this is how these things are measured. Very interesting procedure. So the next thing you have to ask yourself, what vital lessons can we derive from this whole procedure, measurements that we have just discussed? Now, one of the things you need to know is that you need to use what is known to know what is unknown. In other words, use what you have to get what you don't have. That's a very important law and principle in life. So we're not bothered about the fact that we don't know ICF, we don't know ISF. We use the one we knew to get what we can't get normally and directly. Okay? Another lesson is that everything is connected in this life. You might not be able to see the connection, but everything is connected. So what you should aspire to is to be able to gain insight into the connection between things, and then that's how you gain access to whatever you need. Now, the same thing applies in life. Everybody in life is connected. Some people say, oh, I don't have connection and this and that, but it's not true. You know somebody. That person knows another person. That person knows another person. And that's how we are all connected. to gain what we need to gain. All right. So you understand also that without knowing the structure and the relationship between ISF, plasma, you know that diagram we drew, the hearts, arteries, and all of that. Without that knowledge, we will not have been able to know that we can gain access only through the plasma to every. So that means there are foundational knowledge that you need to have in able to achieve certain results. Some people like to jump procedures and processes. Okay, they like to cut corners, and in the end, they fall into very big problems. So you need to start from the foundation. If you have a very poor foundation in anything in life, you are going to fall into problems later. You will crash later. So always go for foundational knowledge. Go for the foundation and build from there. That's how you lead into something complex and very great. All right. So those are the lessons we can learn. And the final lesson that is very important is that measurement of things is one of the keys to controlling, managing, and effectively improving things. So whatever you cannot measure, you cannot control. Whatever you cannot measure, you cannot manage. Whatever you cannot measure, you cannot improve. So take, for example, your blood pressure. You cannot even measure your blood pressure. How can you manage it? Let's say, for example, you have a problem with hypertension. That's high blood pressure. If you cannot measure it, or high glucose level in your blood, if you cannot measure it, you can't control it, you can't manage it. So everything about health, medicine, physiology is to be able to measure so many things in the body so that when it deviates from normal, we know how much correction we need to do to bring it back to normalcy. That's what physiology is all about. That's what medicine is all about. So if you want to control your finances, you must be able to measure your expenditure. So this is a very important lesson. Keep it in your mind. So right now we're going to take a break. When we come back, we'll deal with something very vital.
If you've enjoyed this video so far, kindly click the like button. Do not also forget to subscribe and turn on the notification bell so that you'd be the first to know when we drop a new video every week.
Okay, so whatever you cannot measure, you cannot control, manage, and improve is a very vital lesson you need to learn. So next, we're going to be talking about something also important. I'm sure you have seen pregnant women. Some of them you see their feet swollen. When you press it like this, palpate it, you see some depression. It's called edema. So you ask yourself, how does this thing happen? So we're going to be using the understanding from this body fluid we've discussed to understand the physiological basis of edema and what can be done about it. So we're going to be talking about edema. Okay, the Americans, they spell it like this, edema, or the British, so it's the same thing, edema. So edema generally is the accumulation of excess fluid in some of these compartments of the body, but mainly it usually refers to accumulation of excess tissue fluid. That's the interstitial fluid. Okay, so you have intracellular edema when there's too much fluid in the intracellular compartment. Then the interstitial fluid compartment is part of the extracellular. So that's why we refer to it as extracellular. So you have intracellular edema. You have extracellular edema, and it is in this compartment. Okay, so that's what we're going to be dealing with now. How does fluid accumulate so much in the interstitial fluid compartment that leads to swelling? Okay, So let's revisit that arrangement we talked about before. Okay, so you have tissues like this all around. You have the interstitial fluid in between them. Okay, so you have capillaries from the arterial side, okay, in between. Then halfway in between, you now have the capillaries as towards the venous side. Okay, so this is it. So you have the capillaries, arterial side, capillaries, venous side. So around it, you have interstitial fluid. So there are two major forces that are involved in, the capillaries, they are involved in exchange of fluid. So fluid comes from the capillaries, which is in the intravascular compartment, into the interstitial, and from interstitial into the cell. So that's how the exchange happens. Exchange of oxygen, carbon dioxide, food, nutrients, hormones, everything. This is where the main work happens in the body, the exchange, right? So the two forces we talked about, one of them is called hydrostatic pressure. Hydrostatic pressure. So what is hydrostatic pressure? Hydrostatic pressure is just talking about the water, that's the plasma, the fluid there. The pressure it mounts on the blood vessel that forces fluid from inside the capillaries into the interstitial fluid compartment. Okay, so it's an outside force. So you have a blood vessel like this. Let's say this is a capillary. So, and this is the interstitial space. So it forces it down outside. So this process is known as filtration. Okay, so the hydrostatic pressure leads to filtration. It forces the fluid into this place. So, but there's another thing that happens with the plasma. The plasma, remember we said that it contains plasma proteins, and one of them is that albumin we mentioned, but there are others. So those plasma proteins, they exert a kind of pressure called oncotic pressure. So the pressure, this oncotic pressure is also known as colloid osmotic pressure. So what does this mean? It means that the plasma proteins, they absorb water. Osmosis has to do with the movement of water. So water moves from where it is more to where it is less. That means it moves from where it is more dilute to where there's more concentration. So since you have plasma proteins here, so the fluid here, plasma proteins are not found in the interstitial fluid. It is only found, remember that it cannot pass the capillary membrane. So it draws water from the interstitial fluid compartment back into the capillaries. So you see that it's opposite of this hydrostatic pressure. So these are the two forces and they are opposite each other. So what happens is that at the capillaries, at the arterial side of the capillary, the hydrostatic pressure is about 37 millimeters of mercury, which is taking down fluid, filtering it into the space. Okay, then the oncotic pressure is about 25 millimeters of mercury. So what do you think will happen? Filtration is having 37. This one is called reabsorption. It's taking back the fluid into the vessel. It's called reabsorption. Okay, let's write it here. Reabsorption. So that means filtration is more. It's overriding it. So what is the difference between these two? You have about 12 millimeters of mercury. I would like to state that these figures, they vary depending on the text you read. Okay, some will use even 35. So let's use 35 so that it will be a round figure. So this will now be 10. So the difference between reabsorption and filtration is 10 in favor of filtration at this arterial end. So that means more water gets into this interstitial fluid compartment. So something interesting happens when it now moves to this venous compartment, this venous side. Okay, this one drops. The hydrostatic pressure as it's moving towards the venous side, it's dropping. So at this venous side, it becomes what? 17 millimeters of mercury from 35. But this oncotic pressure, because it cannot cross, so it still remains the same 25. So what happens? The oncotic pressure is now greater than this. So more of reabsorption is happening. So the difference between this and this is what? 8 millimeters of mercury in favor of reabsorption. So the fluid that has filtered will go back. But the interesting thing is that this is 8, this is 10. So there is an imbalance. If you leave it like this, it means more of filtration is happening and more fluid is going to be accumulating here. So what handles this excess of 2 millimeters of mercury is called lymphatic drainage. So also around this place, you have lymphatic vessels that drain the excess fluid and returns it back into the veins. All right, so that's what happens. So we've mentioned 3 important things that contribute to how much fluid is left in this interstitial compartment. We mentioned filtration, we've mentioned reabsorption, and we've mentioned drainage. Lymphatic drainage. 3 things. So, logically, what do you think will lead to excess of interstitial fluid? It means that if hydrostatic pressure is increased beyond normal, that means instead of 35, it goes to 40, 45. That means it's going to be more than 10. So much of filtration is going to predominate and it will cause accumulation of excess fluid, leading to edema. Another thing, reabsorption, oncotic pressure. If oncotic pressure, even if this one remains 35, but oncotic pressure now reduces from 25 to maybe 15, that means you don't have enough plasma proteins. So increase in hydrostatic pressure, then decrease in oncotic pressure can lead to excess accumulation of fluid in the tissues, leading to edema. So the third thing is drainage. So if those lymphatic vessels are blocked, what happens? You also have edema. So we're going to use this understanding to now list the causes of edema. So, 1, increased hydrostatic pressure. A very prominent thing that can cause it is heart failure. So when the heart is not pumping enough blood, distributing it to the system, so there is accumulation. It's not releasing enough. So there's accumulation of blood in the veins. So when it's so accumulated, it shoots up the hydrostatic pressure. So those people who have heart failure, you see them especially at the feet. They have, usually right-sided. The heart is like this. This is the right side. The right side. This is the left side. Okay? So this side is where the veins enter. So this right-sided heart failure manifests a lot as swollen feet. Sometimes it can even reach the laps. When you try to press it, you see the depression. Okay? Prominent thing there, heart failure. Even renal failure too. Because the kidneys is not excreting enough water. So there's accumulation, a lot of water in the body. It leads to edema. So number 2 is decreased oncotic pressure. So decreased oncotic pressure means little plasma protein. You have less plasma proteins in the blood. And what are the things that can lead to that? Number 1 is that you are not taking in enough proteins in your food. So 1 is decreased intake of proteins. A good example is kwashiorkor. Kwashiorkor, those who, it happens to children that have kwashiorkor, they are not taking enough protein in their food. And children with kwashiorkor, their feet, they are swollen, they have edema. Okay? So another thing, apart from decreased intake, you could have loss of proteins that are already there. You are taking enough proteins, but you are losing a lot of proteins through your urine. Normally, proteins are not supposed to be lost in the urine, but now you are losing proteins. So, increased loss of protein. So, if you have kidney disease like glomerulonephritis, it will lead to increased loss of protein. It leads to decreased oncotic pressure and edema. Another one, third one, from this oncotic pressure is decreased manufacturing or synthesis of plasma proteins. Okay, so you have taken in enough proteins, you are not losing proteins, but the liver is not manufacturing and synthesizing enough plasma proteins that you need. So you have decreased synthesis of proteins. So a good example is liver disease. So those who have liver disease, it manifests as edema. There's swelling around their bodies. Okay? So that's oncotic pressure. So the third one is blockage of lymphatic vessels. There's blockage of lymphatic vessels. Something that can cause that. Some parasitic infections. One example is filariasis, caused by filaria worm. There's a worm called filaria worm that when it enters your body, it can block lymphatic vessels. So when it blocks it, that remaining 2 millimeters of mercury that is supposed to be drained by the lymphatic vessels is no longer there. So gradually, it accumulates in the tissues. So there's swelling. Okay? And which leads to elephantiasis. Filariasis can lead to elephantiasis. Infantiasis, people, you've seen some people, one of their limbs is very big, looking like the limb of an elephant. That's why it's called elephantiasis. Okay, so this is how edema comes about. These three things: increased hydrostatic pressure, reduced oncotic pressure, and blockage of the lymphatic vessels. So when you understand this, you see somebody with edema, your mind begins to go, what could be wrong? Is it increased hydrostatic pressure from heart failure, kidney failure, and so on? Is it that the person is not taking enough proteins, especially when it's children? You know what to do. Kidney disease, liver disease. So your mind, this is how the mind of a medical doctor operates. You know the causes because you understand the physiological basis of it, and you are able to do some tests to rule out this one, that one, and then you are able to pinpoint the cause. And when you pinpoint the cause, you can now solve the problem. Right? So this is how edema is formed. We are done with the body fluids. Now is the kidney itself. It looks like a bean, has this bean shape. You are going to be unraveling the structure. You know that structure determines function. So we'll learn one or two things about the structure of the kidney that will lead us to know why the kidney functions the way it functions. Filtering all the waste in the body, excreting them, and leaving the things that are important and removing the ones that are not important. How it does that. We're going to be doing that in the next video. Alright, so for further reading, I've written quite a few books in physiology, about 6 or 7 of them. So you can go and download it. It's on Okada Books. Okada Books. You see the link in the description box. Just download it and read. You see some of these things I've been mentioning. So it will help you get a better grasp of these things.